It's time for the 3-question warrant for Biochem 9. Let's get going. Which cytokines inhibit Th1 cells? That's going to be interleukin 10, and that's going to be what's inhibiting those Th1 cells. Next, outline the flow of aqueous humor. So this formed in a capillary bed in the ciliary body, and that's going to go further on and be secreted into the posterior chamber. Then it flows between the angle formed by the lens and the iris diaphragm. Then it moves into the anterior chamber and is finally reabsorbed by the canal of Schlemm. Next question, what is the mechanism of action of metoclopramide? So this is an antagonist at the dopamine D2 receptor. It's also a serotonin agonist, so it will increase contractility in the GI tract and move things along. All right, that's it for the warm-up. Let's get to that lecture now.
We've covered glycolysis and gluconeogenesis, and there's one more big thing you can do with glucose, which is to store it as glycogen. Now, glycogen is a huge polysaccharide made up of glucose molecules, which is how our hepatocytes and muscle cells store excess glucose for use later when you're fasting and blood glucose levels start to drop. The process of making glycogen is called glycogenesis. So let's look at the glycogenesis pathway in the study guide. Glycogenesis begins with glucose, and you have to trap it in the cell by phosphorylating it, by adding a phosphate group to make glucose 6-phosphate. The two enzymes responsible for that were glucokinase and hexokinase. And do you remember which enzyme was found in which tissues? Glucokinase is found mainly in the liver and the beta cells of the pancreas, and hexokinase is found in other tissues. So if we're talking about glycogenesis in skeletal muscle cells, it starts with hexokinase. So these enzymes turn glucose into glucose 6-phosphate, and then from there, you're going to move the phosphate group from the 6th carbon to the 1st carbon to make glucose 1-phosphate. This is different than glycolysis. With glycolysis, we went from glucose 6-phosphate to fructose 6-phosphate. Here, to synthesize glycogen, we're going from glucose 6-phosphate to glucose 1-phosphate. The next step is to convert it to something called uridine diphosphoglucose, or UDP glucose. And this is catalyzed by an enzyme called UDP glucose pyrophosphorylase. It's going to take that glucose 1-phosphate and make UDP glucose. So it's priming the glucose to be attached to a larger molecule. And the enzyme that attaches UDP glucose to the expanding glycogen molecule is called glycogen synthase. This is the rate-limiting enzyme for glycogen synthesis. Glycogen synthase is going to make what are called alpha-1,4 glycosidic linkages, which make the chain longer and longer and longer. There's another enzyme that makes branches in the glycogen chain, which is called branching enzyme, and it makes alpha-1,6 glycosidic linkages. Glycogen can get to be rather enormous, so the branches help you keep it somewhat compact. So you use alpha-1,4 and alpha-1,6 linkages. So that's glycogen synthesis. The purpose of glycogen is to store glucose for later. When you need glucose, you're going to start breaking down that glycogen and releasing the glucose. And that's a process called glycogenolysis, lysis of glycogen. So take a look at the diagram in the study guide. To break down glycogen, you either have to break down the alpha-1,4 linkage at the top of the diagram, or you have to break down the branching alpha-1,6 linkage at the bottom left. Let's start with the alpha-1,4 linkage. The majority of glycogen is composed of these alpha-1,4 linkages. And the enzyme that's used to break down the alpha-1,4 linkage is called glycogen phosphorylase. So glycogen phosphorylase breaks down the alpha-1,4 linkage and cuts loose one glucose 1-phosphate molecule. Then an enzyme called phosphoglucomutase converts that glucose 1-phosphate to glucose 6-phosphate. And then the enzyme glucose 6-phosphatase converts it back into glucose. Now, where do we see glucose 6-phosphatase before? Well, that's the last step in the gluconeogenesis pathway. And here we see that it's also the last step in glycogenolysis, converting that trapped glucose 6-phosphate back into glucose. Now remember, we said that muscle cells don't have this glucose 6-phosphatase enzyme. That's one reason muscle cells can't perform gluconeogenesis. And it also means that while muscle cells can break glycogen down into glucose 6-phosphate, that glucose 6-phosphate is still trapped in the cell. So it's going to be used by the muscle cell to generate energy through glycolysis. So the only reason a muscle cell breaks down glycogen is for its own use. Muscles can't share energy. Sharing energy is the liver's job, and hepatocytes do have glucose 6-phosphatase. So those are the 1,4 linkages. Now what about the branching linkages, the alpha-1,6 linkages? The enzyme that breaks those down is alpha-1,6 glucosidase, which is also referred to as debranching enzyme. So debranching enzyme, or alpha-1,6 glucosidase, breaks down the alpha-1,6 linkage directly into glucose. You don't have to go through all those other steps with the alpha-1,6 linked glucose. So it's a little bit more efficient. And actually, debranching enzyme has two functions. Let me illustrate it for you. This is your branched glycogen molecule, and glycogen phosphorylase is cooking along, breaking off glucose 1-phosphate molecules one at a time. But when it gets close to a branch point, one of those alpha-1,6 linkages, specifically when it gets exactly 4 glucose molecules away, glycogen phosphorylase stops working. That's when debranching enzyme goes to work. Like I said, debranching enzyme has 2 functions. One function is called a transferase activity. Specifically, it's called 4-alpha-D-glucanotransferase activity. This means that debranching enzyme can take 3 of those last 4 glucose molecules and transfer them to another chain. So transferase transfers the branch to another chain so that glycogen phosphorylase can keep working on it. Then the other function of debranching enzyme is the alpha-1,6 glucosidase activity, which we already discussed. That's how debranching enzyme removes that last glucose molecule. Now let's talk about the regulation of glycogenolysis for a second. We need to regulate the processing of glycogen for the same reasons that we needed to regulate glycolysis and the reasons we needed to regulate gluconeogenesis. Basically, you don't want to use your body's energy to break down glycogen when there's already plenty of glucose in the circulation, such as after a meal. And you don't want to be making glycogen and storing glucose during fasting when the blood glucose is low. You need that glucose for energy. So one regulatory mechanism is that insulin inhibits glycogen phosphorylase and it stimulates glycogen synthase. So when you eat and the bloodstream is flooded with glucose, the pancreas releases insulin to allow that glucose to enter the cells. And at the same time, that insulin ends up inactivating glycogen phosphorylase. So you stop breaking down glycogen. And insulin stimulates glycogen synthase. So you start storing whatever extra glucose you have that you don't need right then. Now, on the other hand, imagine you're in a high stress state, a fight or flight state, where you need to share glucose. So what organ shares glucose? Primarily the liver. So your body communicates a high stress state by releasing epinephrine and glucagon. These hormones are known as counter-regulatory hormones because they do the opposite of insulin. We've already seen that glucagon stimulates adenylyl cyclase in the hepatocytes to make cyclic AMP. That cyclic AMP activates protein kinase A. And we've seen how kinases can switch on certain enzymes by phosphorylating them. So protein kinase A activates glycogen phosphorylase kinase, and that in turn activates glycogen phosphorylase in a roundabout way. Don't get too hung up on the details. The bottom line is that glucagon stimulates glycogen phosphorylase and it stimulates glycogenolysis. That's how glucagon raises the blood sugar. Epinephrine does basically the same thing. Beta-adrenergic receptors are GS receptors, which also stimulate adenylyl cyclase. Remember that GS, the S is for stimulate. And that ends up activating glycogen phosphorylase and again stimulating glycogenolysis. When we discussed glycolysis, we talked about diseases that result from deficiencies of enzymes in the glycolytic pathway, such as pyruvate kinase deficiency. Well, there are also some diseases that result from deficiencies of enzymes of the glycogenolysis pathway. These are collectively called glycogen storage diseases. There are about 11 different glycogen storage diseases, but you only have to know 4 of them. But these 4 are very high yield. The first one I want to discuss is known as McArdle disease, which is a deficiency in glycogen phosphorylase. This is also called glycogen storage disease type 5. With McArdle disease, there's accumulation of excess glycogen in the muscle because all the enzymes that make glycogen are intact, but these patients can't break down glycogen. Specifically, they can't break down those alpha-1,4 linkages in glycogen. So the muscle cells fill up with glycogen, and the osmotic pressure from the glycogen draws water into the cells, and the muscle cells swell up and eventually lyse. And when muscle cells lyse, they spill myoglobin into the serum, and this is a condition called rhabdomyolysis. So that myoglobin goes through the blood to the kidneys, and that causes myoglobinuria, myoglobin in the urine. And the myoglobin can clog up the kidneys and eventually cause renal failure. Now, patients with McArdle disease can also have muscle cramping because their muscles are starved for glucose because they can't break down glycogen. But the good news is that patients with McArdle disease have a normal life expectancy. It's just a problem with the muscle cells. And again, it's because you're deficient in the enzyme glycogen phosphorylase. The next disease is von Gierke disease, or glycogen storage disease type 1. Now, this is a deficiency of glucose 6-phosphatase. And remember, this was the enzyme at the final step of both glycogenolysis and gluconeogenesis. Also remember that skeletal muscle cells don't have glucose 6-phosphatase. So this is a deficiency of glucose 6-phosphatase in those places that would share glucose, such as the liver, the enterocytes, and the kidney. So if these energy-sharing tissues can't perform either gluconeogenesis or glycogenolysis, that means they can't share glucose when the blood glucose starts to drop. So these patients are prone to hypoglycemia when they're fasting. So one characteristic of von Gierke disease is severe fasting hypoglycemia. These patients also have buildup of glycogen in the liver, because again, the liver can make glycogen, it just can't completely break it down. So this causes enlargement of the hepatocytes and hepatomegaly. These patients also get enlargement of the kidneys, they get elevated serum lactate, they get elevated uric acid, and hypertriglyceridemia. And then you can treat von Gierke disease by feeding the patient frequently so that they're going to have a fairly steady stream of dietary glucose to prevent that hypoglycemia. And then at night, you give them cornstarch to prevent overnight hypoglycemia because cornstarch is going to be digested more slowly. The next disease is Cori disease, or glycogen storage disease type 3. Cori disease is like a milder form of von Gierke disease. Cori disease is due to a defect in the debranching enzyme. So these patients can break down the alpha-1,4 linkages. They just can't break down the alpha-1,6 linkages. So glycogen can be broken down, just not completely. And gluconeogenesis works perfectly fine. So these patients have milder hypoglycemia. They have no elevation in lactate levels and no elevation in uric acid. And then the last disease is called Pompe disease, or glycogen storage disease type 2. Pompe disease is caused by a deficiency of the alpha-1,4 glucosidase in the lysosomes. So the lysosomes are responsible for breaking down a small amount of glycogen. So if the muscle cell lysosomes don't have this alpha-1,4 glucosidase, some of the glycogen won't get broken down. Now, there are two forms of Pompe disease. There's an infantile form and an adult form. The infantile form is more severe. These individuals have very, very low levels of enzyme activity, so symptoms appear earlier in infancy. The infantile form of Pompe disease causes severe muscle weakness, cardiomegaly and heart failure, and these patients have a shortened life expectancy, usually less than a year if the disease is left untreated. Then the adult form of Pompe disease, or late onset Pompe disease, occurs when you have some lysosomal enzyme activity, but less than normal. So these patients generally have no cardiac involvement. They get gradual onset of skeletal muscle weakness, as well as diaphragm weakness and respiratory failure. Now it's time for the end of session quiz. So I want you to pause the video, work through those questions, then restart the video and we'll go over the answers. First question, what's the rate limiting enzyme for glycogen synthesis? It's glycogen synthase. And then for glycogenolysis, it's glycogen phosphorylase. So again, make sure you know all those rate limiting enzymes for all these pathways. Next, which enzyme converts glucose 6-phosphate to glucose? It's the enzyme glucose 6-phosphatase. Remember, that's the enzyme that's deficient in von Gierke disease. Last question, which glycogen storage disease matches each of the following phrases? So glycogen phosphorylase deficiency is McArdle disease. Glucose 6-phosphatase deficiency is von Gierke disease. We just mentioned that. Lactic acidosis, hyperlipidemia, and hyperuricemia is von Gierke disease. Alpha-1,6 glucosidase deficiency, that's debranching enzyme, which is deficient in Cori disease. Alpha-1,4 glucosidase deficiency is Pompe disease. Cardiomegaly is seen with Pompe disease, the infantile type. And then diaphragm weakness and respiratory failure is adult type Pompe disease. Increased glycogen in the liver and severe fasting hypoglycemia is von Gierke disease. Hepatomegaly, hypoglycemia, hyperlipidemia, but with normal kidneys, normal lactate, and normal uric acid is Cori disease. Painful muscle cramps and myoglobinuria with strenuous exercise is McArdle disease. And severe hepatosplenomegaly with enlarged kidneys is von Gierke disease. So if you know nothing else about these diseases, know those bullet points. Very high yield. And that's it for now. I'll see you next time.